Welcome to our channel. We are so glad you found your way here. Let us help you fall asleep. Let's begin with photosynthesis, the alchemy of turning light into life. And to life, take a slow, deep breath in and release it. Let the weight of the day fade away as we embark on an unhurried, atmospheric journey through the wonders of the cosmos. Your mind is quiet, the universe is vast, and we are ready to explore. Photosynthesis serves as a fundamental process by which green plants, algae, and certain bacteria convert light energy into chemical energy. This transformation initiates with the absorption of sunlight primarily by chlorophyll, the green pigment found in chloroplasts. This region of the cell acts as a site where light energy is harnessed, driving the conversion of carbon dioxide and water into glucose and oxygen. Light-dependent reactions occur first, utilizing the energy from sunlight to split water molecules, releasing oxygen as a byproduct. This energy is captured in the form of adenosine triphosphate and nicotinamide adenine dinucleotide phosphate, essential molecules that fuel subsequent processes. The Calvin cycle follows, where atmospheric carbon dioxide is fixed into organic molecules. Without this process, the planet would be devoid of the rich diversity of life that thrives today. The products of photosynthesis not only sustain the organisms that perform it, but also serve as a foundational energy source for countless others. This interplay reflects the interconnectedness of ecosystems, illustrating the profound significance of photosynthesis within the biosphere. Its quiet yet powerful role continues to shape the evolutionary trajectory of life on Earth. This process serves as the foundation for most ecosystems, underpinning the intricate web of life that flourishes in diverse environments. Through the conversion of solar energy into chemical energy, it allows autotrophic organisms such as plants and algae to produce organic compounds. These compounds become essential nutrients for a multitude of heterotrophic organisms, from the smallest herbivores to apex predators. In terrestrial ecosystems, forests and grasslands thrive due to the availability of sunlight, which fuels photosynthesis. The resulting biomass not only supports various species but also contributes to soil formation and stabilization. In aquatic environments, phytoplankton, microscopic organisms that perform photosynthesis, play a pivotal role in carbon cycling and oxygen production. They are, in fact, responsible for generating a significant portion of the oxygen present in the atmosphere. This connection between photosynthesis and energy flow highlights its critical role in sustaining biodiversity. As energy transitions through different trophic levels, it maintains the balance of ecosystems. Any disruption in this delicate process can lead to cascading effects, threatening the stability of entire habitats. The ongoing study of photosynthesis reveals its complexities, including adaptations that allow organisms to optimize light capture in varying conditions. Understanding these mechanisms is essential for addressing challenges such as climate change and food security. Pioneering investigations into photosynthesis began in the 17th century, when scientists like Jan Baptist van Helmont conducted experiments that revealed the relationship between plants and water. These early inquiries laid the groundwork for subsequent discoveries regarding plant growth and its dependency on sunlight. In the 19th century, the work of Joseph Priestley and Jan Ingenhousz illuminated the role of light in the process, demonstrating that plants release oxygen, The advent of the 20th century marked a significant turning point in our comprehension of this vital process. The elucidation of the chlorophyll molecule by Richard Willstetter provided critical insights into how light energy is captured. Following this, researchers like Melvin Calvin further unraveled the biochemical pathways, detailing the intricate series of reactions that convert carbon dioxide and water into glucose. This exploration, known as the Calvin cycle, underscored the complex interplay between light and life. Advancements in technology during the late 20th century, including the use of radioactive isotopes, allowed for the visualization of photosynthetic processes at an unprecedented scale. These innovations expanded understanding, revealing the minute details of energy transfer and the significance of various pigments and enzymes within chloroplasts. As this field continues to evolve, contemporary research is increasingly focused on enhancing photosynthetic efficiency to combat global challenges and harness renewable energy sources. Key contributors to photosynthesis science have shaped the understanding of this essential process over centuries. Notable figures include Jan Ingenhouser, who in the 18th century discovered that plants absorb carbon dioxide and release oxygen in the presence of sunlight. His observations laid crucial groundwork, revealing the significance of light in plant metabolism. Subsequent advancements came from Joseph Priestley, whose experiments illustrated the concept of gaseous exchange between plants and their environment. This foundational work provided insights into the role of carbon dioxide in photosynthesis. The early 20th century saw the contributions of Melvin Calvin, whose research elucidated the intricate biochemical pathways of carbon fixation, now known as the Calvin cycle. This process is integral to converting carbon dioxide into organic compounds. Further developments emerged from the elucidation of the photosystems, particularly the work of Robert Hill and others. Their investigations into the light-dependent reactions expanded knowledge of how chlorophyll captures sunlight, driving the conversion of light energy into chemical forms. The integration of molecular biology and genetics has revolutionized research, allowing scientists to manipulate and enhance photosynthetic pathways. These contributions collectively unveil the complexity of photosynthesis, enhancing the understanding of plant biology and its applications in agriculture and renewable energy. Each discovery builds upon the last, forming a cohesive narrative of scientific progress in this vital area. The fundamental equation of photosynthesis exemplifies the intricate conversion of light energy into chemical energy. In its simplest form, this process can be represented as 6 molecules of carbon dioxide and 6 molecules of water, yielding 1 molecule of glucose and 6 molecules of oxygen. This transformation occurs primarily within the chloroplasts of plant cells, where chlorophyll captures sunlight. Light energy instigates a series of reactions that the significance of this equation extends beyond mere numbers. It encapsulates the essence of energy flow within ecosystems. Plants act as primary producers, converting solar energy into a form that can be utilized by various organisms. This interaction is foundational, as it underpins the survival of herbivores, which in turn sustain carnivores. The efficiency of photosynthesis is influenced by various factors, including light intensity, temperature, and carbon dioxide concentration. Understanding this delicate balance reveals not only the complexity of biological systems, but also the critical role plants play in maintaining atmospheric stability and supporting life. Light serves as the primary energy source for the process of photosynthesis. initiating a series of intricate biochemical reactions within chloroplasts. In this region of green leaves, chlorophyll absorbs sunlight, converting it into chemical energy. This absorption occurs predominantly in the blue and red wavelengths, enabling plants to harness energy efficiently from the spectrum of sunlight. Once inside the chloroplasts, the absorbed light energy excites electrons, propelling them through a series of protein complexes known as the electron transport chain. This cascade creates a proton gradient, facilitating the synthesis of adenosine triphosphate, or ATP, which acts as a crucial energy currency within cells. Simultaneously, the absorbed energy drives the conversion of carbon dioxide and water into glucose and oxygen through a series of reactions known as the Calvin cycle. This process not only generates organic compounds vital for plant growth and development, but also releases oxygen as a byproduct, enriching the atmosphere. The significance of light in photosynthesis extends beyond the confines of individual plants. It underpins entire ecosystems, influencing food webs and the carbon cycle. The interplay between light, plants, and the atmosphere sustains life on Earth, demonstrating the profound interconnectedness of environmental systems. This relationship underscores the importance of light as an essential driver of biological productivity and atmospheric health. Understanding the electromagnetic spectrum reveals the breadth of light's influence on life, spanning a range of wavelengths. This spectrum encompasses various types of radiation, from radio waves to gamma rays, visible light. A narrow band within this continuum plays a critical role in photosynthesis. Chlorophyll, the pigment found in plants, absorbs specific wavelengths, primarily in the blue and red regions, enabling this life-sustaining process. The energy harnessed from light triggers a series of chemical reactions within chloroplasts. This energy conversion transforms carbon dioxide and water into glucose and oxygen, signifying a fundamental biochemical pathway. In doing so, it not only fuels individual organisms, but also supports entire ecosystems, creating a foundation for complex food webs. Beyond the visible spectrum, ultraviolet radiation poses both challenges and opportunities. While it can cause cellular damage, certain organisms have evolved mechanisms to utilize this energy, further diversifying the spectrum of life on Earth. Infrared radiation, with its heat-carrying capacity, influences plant growth patterns and water cycles, shaping habitats and ecosystems. The intricate interplay of these wavelengths illustrates the interconnectedness of life and light. Variations in solar radiation can profoundly affect climate and environmental conditions, highlighting the delicate balance that sustains life in this region and beyond. Understanding this spectrum enhances comprehension of both terrestrial and extraterrestrial biological processes. Photosynthetic pigments play a crucial role in capturing light energy, with chlorophyll being the most prominent among them. This pigment absorbs light primarily in the blue and red wavelengths, reflecting green, which is why many plants exhibit this hue. Chlorophyll's structure allows it to efficiently absorb photons, initiating a series of reactions that convert light energy into chemical energy. Other pigments, such as carotenoids and phycobilins, complement chlorophyll by broadening the absorption spectrum. Carotenoids, for instance, capture light in the blue-green spectrum and protect against photooxidative damage. These pigments are responsible for the vibrant yellows and oranges seen in many fruits and vegetables. contributing not only to photosynthesis, but also to the visual diversity of the biosphere. In aquatic environments, phycobilins serve a similar purpose, allowing certain algae to thrive in deeper waters where light is scarce. This adaptation enables them to harvest energy from wavelengths that penetrate more efficiently into the depths of oceans and lakes. The interplay among these pigments enhances the overall efficiency of photosynthesis, enabling a wide range of organisms to harness solar energy.
As research advances, the understanding of these pigments continues to evolve, revealing intricate mechanisms that have sustained life on Earth and potentially beyond. Light absorption initiates the complex process of photosynthesis. Within chloroplasts, pigments like chlorophyll capture photons, exciting electrons, and creating an energy-rich state. This energy facilitates the transformation of carbon dioxide and water into glucose, a fundamental building block for life. The absorption of light occurs primarily in the blue and red wavelengths, highlighting the efficiency of these pigments in harnessing solar energy. Energy transfer unfolds through a series of molecular interactions, where excited electrons travel through a network of proteins and pigments. This journey occurs within the thylakoid membranes, where they are passed along, gradually losing energy. Each transfer is highly orchestrated, ensuring that the maximum amount of energy is captured and utilized. In the process, the energy is used to pump protons into the thylakoid lumen, generating a proton gradient essential for ATP synthesis. The resulting adenosine triphosphate serves as the cellular currency for energy. Following this, the electrons are eventually transferred to NADP+, forming NADPH, a crucial cofactor in the subsequent reactions of photosynthesis. This intricate energy transfer mechanism illustrates the profound efficiency of life systems. The evolution of these processes not only sustains terrestrial ecosystems, but also hints at the potential for similar mechanisms elsewhere in the universe, where light may drive the emergence of life. Chloroplasts serve as the essential sites of photosynthesis within plant cells, embodying a complex yet elegantly organized structure. Enclosed by a double membrane, these organelles house an intricate arrangement of thylakoids, which are flattened, disc-like sacs stacked into structures known as grana. This arrangement maximizes the surface area available for the crucial light-dependent reactions. Each thylakoid membrane is imbued with chlorophyll, the pigment responsible for capturing sunlight, transforming it into chemical energy. Surrounding the thylakoids is the stroma, a gel-like fluid where the light-independent reactions, also referred to as the Calvin cycle, occur. Within this region, carbon dioxide is fixed into organic molecules, demonstrating a remarkable transformation of energy and matter. This precise orchestration of components facilitates the conversion of solar energy into glucose. The evolution of chloroplasts showcases endosymbiotic theory, suggesting that these organelles originated from free-living cyanobacteria that were engulfed by ancestral eukaryotic cells. This symbiotic relationship highlights the intricate interconnectedness of life forms and their environments. The chloroplast's ability to harness light energy not only enriches the cells they inhabit, but also plays a foundational role in sustaining the broader biosphere. As photosynthesis continues to unfold, it remains a cornerstone of ecological balance, driving the cycles of energy and nutrients essential for life on Earth. The intricate structure of thylakoid membranes plays a pivotal role in the photosynthetic process. This region, found within chloroplasts, consists of stacked, disc-like formations known as grana, interconnected by stroma thylakoids. The arrangement maximizes surface area, allowing for an efficient capture of light energy essential for the conversion of carbon dioxide and water into glucose and oxygen. Proteins embedded within these membranes facilitate the absorption of light and the subsequent transfer of electrons. These proteins include various pigments, such as chlorophyll, which are responsible for harnessing sunlight. The specific wavelengths of light absorbed by chlorophyll initiate a series of reactions known as the light-dependent reactions. This phase occurs predominantly in the thylakoid membranes, where light energy is converted into chemical energy in the form of adenosine triphosphate and nicotinamide adenine dinucleotide phosphate. Beyond energy conversion, the thylakoid membranes also play a crucial role in creating a proton gradient across their structure. This gradient drives the synthesis of adenosine triphosphate through ATP synthase, a process vital to sustaining cellular functions. The regulation and maintenance of this functional architecture enable the efficient operation of photosynthesis, ensuring that energy transfer occurs seamlessly within the cell. This precise organization reflects nature's ability to optimize processes for the sustenance of life. The stroma serves as the matrix of chloroplasts, a fluid-filled space that plays a critical role in the photosynthetic process. Within this environment, enzymes and substrates interact to facilitate the conversion of carbon dioxide and water into glucose. The surrounding thylakoids house chlorophyll, capturing sunlight and initiating the series of reactions necessary for energy transformation. This region supports the Calvin cycle, where carbon fixation takes place. In this cycle, carbon dioxide molecules are incorporated into organic compounds. Chloroplasts, located within plant cells, are essential for harnessing solar energy. They provide not only energy but also oxygen as a byproduct, a critical element for aerobic organisms. The stroma, often overlooked, is where the magic of photosynthesis truly manifests, enabling life to flourish in various ecosystems. As carbon compounds are synthesized, they serve as building blocks for other essential biomolecules, reinforcing the interconnectedness of life forms. This complex biochemical interplay underscores the stroma's importance, highlighting its role in sustaining the delicate balance of terrestrial ecosystems. These processes contribute to the overall health of our planet, illustrating the profound impact of microscopic structures on global systems. Water serves as a crucial component in the intricate process of photosynthesis. It not only acts as a solvent, but also participates directly in the biochemical reactions that transform light energy into chemical energy. This molecule is absorbed by plant roots and transported through the vascular system, reaching the leaves where photosynthesis occurs. Within the chloroplasts, the site of photosynthesis, water molecules undergo a process known as photolysis. This reaction involves the splitting of water into oxygen, protons, and electrons when exposed to light. Oxygen is released as a byproduct, contributing to the atmosphere and benefiting aerobic life forms. The protons and electrons generated during photolysis play a significant role in the formation of adenosine triphosphate, or ATP, and nicotinamide adenine dinucleotide phosphate, or NADPH. These energy-rich molecules are essential for the subsequent stage of photosynthesis, known as the Calvin cycle, where carbon dioxide is fixed into organic compounds. In addition to its chemical functions, water helps regulate temperature within plants, maintaining cellular homeostasis. It facilitates nutrient transport and supports structural integrity. The availability of water directly influences photosynthetic efficiency, highlighting its importance in ecosystems. Thus, the role of this simple molecule extends far beyond mere hydration, underscoring its pivotal position in sustaining life on Earth. The significance of carbon dioxide in the process of photosynthesis cannot be understated. This gas, present in trace amounts in the atmosphere, serves as an essential building block for the creation of organic matter. In the presence of sunlight, It enters the leaf through tiny openings called stomata, where it undergoes a remarkable transformation, a thawelth, within chloroplasts. Carbon dioxide combines with water, initiating a series of reactions that is, in this delicate balance. Carbon dioxide is both a contributor and a regulator of the global climate. Elevated concentrations in the atmosphere can enhance photosynthetic activity, leading to increased plant growth. Conversely, excessive levels can result in climate change, impacting ecosystems worldwide. This duality highlights the complexity of interactions within this system. Plants, through their efficient use of carbon dioxide, maintain the equilibrium necessary for life, while the surrounding environment continuously influences their growth and distribution. This intricate relationship underscores the vital role of carbon dioxide in sustaining life on our planet. Light-dependent reactions initiate the photosynthetic process, occurring within the thylakoid membranes of chloroplasts. Sunlight triggers the absorption of photons by chlorophyll molecules, a pigment responsible for the distinctive green color of plants. This energy causes the excitation of electrons, which are then transferred through a series of proteins known as the electron transport chain. As electrons move along this pathway, energy is harnessed to pump protons into the thylakoid lumen, creating a proton gradient. This gradient drives the synthesis of adenosine triphosphate, or ATP, and NADPH, essential energy carriers for the subsequent phase. Transitioning into the light-independent reactions, often referred to as the Calvin cycle. These processes take place in the stroma of chloroplasts. ATP and NADPH, produced in the light-dependent reactions, fuel the conversion of carbon dioxide into organic compounds. Through a series of enzymatic reactions, carbon dioxide is fixed into 3 carbon molecules, eventually forming glucose and other carbohydrates. This transformation not only supports the organism's energy needs, but also contributes to the complex web of life by providing energy to herbivores and, subsequently, to carnivores. Understanding these stages clarifies the remarkable efficiency of plants in capturing and converting solar energy, highlighting the delicate balance that sustains ecosystems across the globe. This intricate process begins within the thylakoid membranes of chloroplasts, where light energy is harnessed. It involves 2 distinct photosystems, each playing a critical role in the conversion of photons into chemical energy. Upon absorption of light, chlorophyll molecules become excited, initiating a series of reactions that lead to the movement of electrons through the electron transport chain. This movement generates a proton gradient across the thylakoid membrane as protons flow back into the stroma through ATP synthase. The enzyme catalyzes the phosphorylation of adenosine diphosphate into adenosine triphosphate, commonly known as ATP. This chemiosmotic mechanism demonstrates the efficiency with which light energy is transformed into a usable form. Accompanying this process, the reduction of nicotinamide adenine dinucleotide phosphate occurs, capturing high-energy electrons. These electrons are critical for the subsequent synthesis of glucose during the Calvin cycle. The entire photophosphorylation process underlines the remarkable adaptability of plants, allowing them to thrive in various environments through efficient energy capture and conversion. The implications of this mechanism extend beyond individual plants. It influences global carbon cycles and contributes to the atmospheric balance vital for sustaining life on Earth. Each step in this process reflects a finely tuned system, essential for maintaining ecological equilibrium. Light-dependent reactions occur primarily within the thylakoid membranes of chloroplasts, where they capture sunlight, transforming it into chemical energy. This region becomes a site of intricate biochemical activity. Chlorophyll molecules absorb photons, initiating a series of electron transfers that propel the process forward. Excited electrons travel through a chain of proteins known as the electron transport chain. As they move, energy is released, facilitating the pumping of protons into the thylakoid lumen. This creates a proton gradient, which is essential for the synthesis of adenosine triphosphate, or ATP. ATP serves as a vital energy currency for the cell. Simultaneously, water molecules are split in a process called photolysis, releasing oxygen as a byproduct. This reaction also provides electrons to replace those lost by chlorophyll, further sustaining the cycle. The resulting protons and electrons contribute to the formation of nicotinamide adenine dinucleotide phosphate, or NADPH, another crucial energy carrier. These light-driven transformations occur within specialized structures known as photosystems. Photosystem I and Photosystem II are integral to the photosynthetic process, each playing distinct yet complementary roles. Photosystem II initiates the journey of energy capture, harnessing photons to excite electrons from water molecules. This reaction results in the splitting of water, releasing oxygen as a byproduct, while generating a flow of electrons that powers the synthesis of adenosine triphosphate, or ATP. In contrast, Photosystem I operates later in the sequence, collecting electrons energized by sunlight and further elevating their energy state. This complex then facilitates the reduction of NADP+ to NADPH, completing the energy conversion cycle. The orchestration of these two systems illustrates a finely tuned process of energy transfer, underpinned by intricate protein structures and pigments that absorb specific wavelengths of light. The arrangement of these complexes within the thylakoid membranes creates microenvironments ideal for efficient energy transfer. Light-harvesting complexes capture incoming photons, funneling energy to the reaction centers of both systems. These interactions highlight the remarkable efficiency and adaptability of photosynthetic organisms, allowing them to thrive in diverse environments. The interplay between Photosystem I and II exemplifies a fundamental biological process, sustaining life on Earth through the transformative power of sunlight. The Z-scheme of electron transport outlines the intricate series of reactions within the thylakoid membranes. This pathway begins with the absorption of photons by chlorophyll molecules, initiating the excitation of electrons. These energized particles are then transferred through a series of protein complexes and mobile carriers embedded in the thylakoid structure. Initial energy input elevates electrons to a higher energy state, enabling them to traverse the electron transport chain. As they move, they release energy, which is harnessed to pump protons across the thylakoid membrane, creating a proton gradient. This gradient serves as potential energy, later utilized by ATP synthase to produce adenosine triphosphate, a vital energy currency for cellular processes. Chlorophyll replenishes lost electrons by splitting water molecules, a reaction releasing oxygen as a byproduct. This essential step not only sustains the electron flow, but also contributes to the atmospheric composition, maintaining a delicate balance. The journey culminates in the reduction of NADP+ to NADPH, providing the reducing power necessary for the subsequent stages of photosynthesis. The Z-scheme illustrates the interconnectedness of light capture and energy conversion, an elegant mechanism that underpins the survival of numerous organisms. Each component, from light absorption to energy storage, plays a critical role in sustaining the biological fabric of life on this planet. The energy conversion process hinges on a remarkable enzyme known as ATP synthase. Situated within the inner mitochondrial membrane, this molecular machine facilitates the synthesis of adenosine triphosphate, or ATP, the primary energy currency of the cell. It operates through a mechanism that harnesses a proton gradient established by the electron transport chain. As protons traverse the enzyme, they induce a rotational motion that catalyzes the conversion of adenosine diphosphate and inorganic phosphate into ATP. This enzymatic action occurs with astonishing efficiency, as ATP synthase can produce approximately 100 molecules of ATP per second. The intricacies of its structure reveal an elegant assembly of subunits that work in unison. The catalytic sites, where ATP production occurs, are located on the enzyme's stalk, while a ring of C subunits forms the rotor, spinning in response to proton flow. The implications of ATP synthase extend beyond mere energy production. This process is fundamental to cellular respiration and, by extension, to the functioning of all aerobic organisms. It is a critical pivot in the chain of energy transformation, linking the energy harnessed from food with the biological processes that sustain life. These interactions illustrate the profound interconnectedness of biochemistry and the energy dynamics that govern existence on Earth. Nicotinamide adenine dinucleotide phosphate, commonly known as NADPH, serves as a crucial electron carrier within the photosynthetic process. This molecule participates directly in the light-dependent reactions, facilitating the conversion of solar energy into chemical energy. The photons absorbed by chlorophyll excite electrons, which are subsequently transferred through a series of proteins embedded in the thylakoid membrane. This transfer generates a proton gradient, leading to the synthesis of adenosine triphosphate, or ATP. Simultaneously, the excited electrons reduce NADP+ to form NADPH, effectively capturing energy in a stable form. This transformation embodies the transition from light to chemical energy, a fundamental process that sustains the majority of life on the planet. The resulting NADPH and ATP are then utilized in the Calvin cycle, where carbon dioxide is fixed into organic molecules. In this cycle, NADPH donates electrons, contributing to the reduction of 3-phosphoglycerate into glyceraldehyde 3-phosphate, a precursor for glucose and other carbohydrates. The delicate balance of these reactions underscores the efficiency of photosynthesis, allowing plants to harness sunlight while producing oxygen as a byproduct. This intricate mechanism not only sustains plant life, but also underpins the energy resources that support myriad organisms across diverse ecosystems. The Calvin cycle serves as a crucial component within the broader framework of photosynthesis, occurring in the stroma of chloroplasts. This cycle operates through a series of biochemical reactions that convert carbon dioxide into glucose. It begins with the fixation of carbon dioxide, where the enzyme ribulose bisphosphate carboxylase/oxygenase, commonly known as RU-BISCO, catalyzes the reaction between carbon dioxide and ribulose bisphosphate. The resulting 3-carbon compound undergoes a transformation into 3-phosphoglycerate. Subsequent steps involve the reduction of 3-phosphoglycerate into glyceraldehyde 3-phosphate, utilizing adenosine triphosphate and nicotinamide adenine dinucleotide phosphate, which are generated during the light-dependent reactions. This process highlights the seamless interplay between light capture and the synthesis of organic compounds. Not all synthesized glyceraldehyde 3-phosphate molecules are utilized for immediate energy. Instead, some are redirected to regenerate ribulose bisphosphate, ensuring the cycle's continuation. This regeneration requires additional energy inputs, further emphasizing the interconnected nature of these metabolic pathways. The Calvin cycle operates continuously, adapting to varying concentrations of carbon dioxide and light availability. This adaptability illustrates the resilience of photosynthetic organisms, enabling them to thrive in diverse environments. Understanding this cycle reveals the extraordinary efficiency with which life captures and transforms energy within the biosphere. This process begins with carbon fixation, a fundamental step in the Calvin cycle that occurs within the stroma of chloroplasts. It initiates with the enzyme ribulose bisphosphate carboxylase/oxygenase, commonly known as RU-BISCO. This enzyme catalyzes the reaction between carbon dioxide and ribulose bisphosphate, resulting in a highly unstable 6-carbon intermediate. This intermediate quickly breaks down into 2 molecules of 3-phosphoglycerate, marking the conversion of inorganic carbon into an organic form. The significance of this reaction extends beyond mere carbon capture. It serves as a foundation for the synthesis of glucose and other carbohydrates, essential for the energy needs of plants and ants, by extension, the entire food web. This transformation exemplifies the interconnectedness of life. As the absorbed carbon dioxide, light-dependent reactions occurring earlier provide the necessary ATP and NADPH for subsequent stages. The process reflects the delicate balance within ecosystems, as carbon fixation not only supports plant life, but also influences atmospheric composition and climate regulation. The intricate workings of this cycle underscore the remarkable adaptability of organisms to utilize available resources. Each turn of the cycle illustrates the profound elegance with which nature orchestrates energy flow, fostering life across diverse environments. This intricate process relies heavily on ribulose 1,5-bisphosphate, commonly referred to as RuBP. This 5-carbon molecule serves as a crucial substrate in the initial stages of carbon fixation. Present within the chloroplasts, it interacts with carbon dioxide, facilitating the transformation of inorganic carbon into organic compounds. The enzyme ribulose bisphosphate carboxylase/oxygenase, often abbreviated as RuBusco, catalyzes this pivotal reaction. The formation of 2 molecules of 3-phosphoglycerate from each RuBP molecule marks the beginning of the Calvin cycle, a series of biochemical reactions essential for synthesizing sugars. This transformation occurs within the stroma of chloroplasts, where the environment is conducive to such biochemical activities. As carbon dioxide diffuses into leaf tissues, it encounters RuBP, engaging in a series of exchanges that, in ecosystems, the abundance of RuBP contributes to the flourishing of various plant species, which in turn supports a wide array of life forms. This fundamental molecule, while seemingly simple, plays an outsized role in sustaining the biosphere, highlighting the delicate balance of life's processes within this region. The cycle continues, ever reliant on the quiet efficiency of RuBP. The primary enzyme facilitating this transformative process is RuBisco, which catalyzes the reaction between carbon dioxide and ribulose bisphosphate. This critical enzyme plays a vital role in fixing carbon, a fundamental step in the Calvin cycle. Through its action, it enables the conversion of inorganic carbon into organic compounds, a crucial mechanism sustaining life on Earth. RuBusco exhibits dual activity, functioning both as a carboxylase and an oxygenase. In its carboxylase role, it efficiently facilitates the incorporation of carbon dioxide into organic molecules. Conversely, its oxygenase activity leads to a competing reaction that results in photorespiration, a process less favorable for energy production. This duality reflects the delicate balance within photosynthesis, where environmental conditions dictate its efficiency. The presence of RuBisco in the stroma of chloroplasts marks its significance. It is here that the enzyme engages with substrate molecules, initiating a complex series of reactions that, as sunlight bathes the leaves, this enzyme tirelessly works to sustain the energy needs of the vast array of life forms, maintaining an intricate equilibrium within ecosystems. The subtle interplay between these biochemical processes underlines the elegance of nature's design. The stage of photosynthesis involves the reduction phase, where 3-phosphoglycerate undergoes transformation into glyceraldehyde 3-phosphate, commonly known as G3P, utilizing the energy stored in adenosine triphosphate and the reducing power of nicotinamide adenine dinucleotide phosphate. This region witnesses a remarkable chemical conversion in the chloroplasts. Enzymes facilitate the reaction, allowing the phosphorylated compounds to undergo a series of transformations. The presence of a reducing agent plays a crucial role. Reducing agents donate electrons, effectively driving this reduction process forward. The result is a simple sugar that serves as a fundamental building block for organic compounds. This molecular transition not only exemplifies the efficiency of cellular metabolism, but also highlights the interconnectedness of various pathways within the plant cell. Through these transformations, G3P emerges as a versatile molecule, capable of entering multiple biosynthetic routes. It can contribute to the formation of glucose, starch, and other essential carbohydrates. As G3P accumulates, it signals the completion of a critical phase in the photosynthetic process, enabling the plant to harness and store energy derived from sunlight. The quiet progression of this transformation reflects the meticulous and orderly nature of biochemical processes that sustain life on Earth. The regeneration phase marks a pivotal moment in the photosynthetic process, where ribulose biphosphate, or RuBP, is meticulously reformed. This occurs in the stroma of chloroplasts, where the Calvin cycle continues its intricate operation. The presence of enzymes such as Rubusco facilitates the conversion of 3-phosphoglycerate back into RuBP, utilizing energy derived from adenosine triphosphate and reducing power from nicotinamide adenine dinucleotide phosphate. In this region, the cycle's efficiency is paramount, as it ensures the continuity of carbon fixation and the synthesis of organic compounds essential for growth and development. Each cycle yields an output that is carefully controlled, maintaining a balance within the cellular environment. The regeneration of RuBP is not merely a replenishing act. It serves as a link connecting the fixation of carbon dioxide to the eventual production of glucose and other carbohydrates, which are fundamental energy sources for living organisms. The entire process is governed by a series of feedback mechanisms, ensuring that when carbon dioxide levels rise, the cycle accelerates, while a decrease triggers a corresponding slowdown. This finely tuned system exemplifies nature's resilience and adaptability, allowing ecosystems to thrive in various conditions. Through these biochemical reactions, life continues to flourish, sustained by a relentless interplay of energy, matter, and molecular ingenuity. Light intensity serves as a primary factor influencing the rate of photosynthesis. Within this intricate process, chlorophyll absorbs photons, initiating the conversion of light energy into chemical energy. Optimal light conditions enable plants to produce glucose at an accelerated pace, supporting growth and vitality. As intensity increases, the rate of photosynthesis also rises until reaching a plateau where other limiting factors become significant. In lower light conditions, the efficiency of energy capture diminishes. Leaves may exhibit a range of adaptations, such as larger surface areas or a greater concentration of chlorophyll, allowing them to maximize energy absorption. These adaptations reveal the dynamic relationship between organisms and their environments, illustrating how life responds to varying light availability. Beyond mere presence, the quality of light plays a crucial role. Different wavelengths affect photosynthetic efficiency differently, with blue and red light being the most effective for chlorophyll absorption. In shaded areas or under a canopy, plants often compete for limited light, leading to diverse growth strategies. Seasonal changes also impact light intensity. During spring and summer, longer days allow for increased photosynthetic activity, while autumn's shorter days signal a shift in energy allocation. Understanding these factors provides insight into how ecosystems function and adapt to their environments, revealing the delicate balance of life supported by light. Temperature plays a critical role in the efficiency of photosynthesis, influencing the enzymatic processes that drive this essential reaction. In this region, variations in temperature affect the rate at which plants convert light energy into chemical energy. Optimal temperatures enhance enzyme activity, facilitating the transformation of carbon dioxide and water into glucose and oxygen. At lower temperatures, metabolic processes slow, resulting in reduced rates of photosynthesis. Plants exhibit stress, and their ability to grow and reproduce diminishes. Conversely, excessive heat can lead to denaturation of enzymes, impairing their function and causing irreversible damage to cellular structures. These fluctuations create a spectrum of responses among different species. Some thrive in cooler climates, utilizing the lower thermal energy efficiently, while others are adapted to withstand higher temperatures, maximizing their photosynthetic output in warmer environments. In regions experiencing climate change, a to under higher firewalls, the resulting shifts in temperature patterns can lead to significant disruptions. Many flora and fauna may struggle to adapt to rapid changes, potentially leading to altered community dynamics and ecosystem structures. The interplay between temperature and photosynthesis serves as a fundamental aspect of ecological resilience. This relationship highlights the intricate dependencies within ecosystems, where even minor shifts can reverberate through food webs and biodiversity, shaping the future of life on Earth. Unavailability of water serves as a fundamental factor in the process of photosynthesis. This vital resource is absorbed through the roots of plants and transported to leaves, where chemical reactions occur. Insufficient water supply can lead to a decrease in photosynthetic efficiency, directly impacting growth and productivity. Stomata, small openings on leaf surfaces, regulate water loss and gas exchange, balancing the need for carbon dioxide intake with the risk of dehydration. In arid regions, plants have adapted to maximize water use, employing mechanisms such as reduced leaf surface area or specialized pathways to conserve moisture. The resilience of these organisms is critical in maintaining ecological balance, especially under changing climatic conditions that may lead to prolonged droughts. Groundwater levels and surface water availability are essential considerations in agricultural practices, influencing crop yields and food security. Irrigation methods aim to optimize water distribution, yet they must be balanced with conservation efforts to prevent depletion of local water sources. Variations in precipitation patterns can trigger cascading effects within ecosystems, impacting not just photosynthesis, but also soil health and nutrient cycling. This interconnectedness emphasizes the need for sustainable management practices that consider the delicate relationship between water resources and plant vitality, reflecting the overall health of terrestrial environments. The impact of carbon dioxide concentration presents a critical aspect of photosynthesis. Elevated levels of this gas influence the rate at which plants convert light energy into chemical energy. In environments where carbon dioxide concentrations rise, a notable increase in photosynthetic activity occurs, leading to enhanced growth rates for many species. This phenomenon plays a significant role in the global carbon cycle, as plants absorb carbon dioxide, mitigating some effects of climate change. The relationship between carbon dioxide and plant health is complex. While higher concentrations can stimulate growth, they can also lead to imbalances in nutrient availability. Soil health, often intertwined with plant vitality, can suffer under these conditions, as the increased growth may outpace the soil's ability to provide essential nutrients. This region's ecosystems may experience shifts in species composition as certain plants thrive under altered conditions while others decline. The implications extend beyond individual communities. As photosynthesis rates increase, so too does the potential for altered atmospheric conditions. The interplay of carbon dioxide, light, and water creates an intricate web of dependencies. These interactions dictate not only plant growth, but also the overall resilience of ecosystems. Understanding this dynamic is essential for predicting future responses to climate variations and for developing informed conservation strategies in an ever-changing world. Photosynthesis varies significantly across environments, with aquatic and terrestrial systems exhibiting distinct adaptations in aquatic habitats. Light penetration diminishes with depth, leading to a reliance on specific wavelengths that facilitate photosynthesis. Phytoplankton, microscopic organisms inhabiting the upper layers of oceans, utilize chlorophyll to capture sunlight. These organisms are responsible for a substantial portion of global oxygen production, thriving in nutrient-rich waters. The efficiency of photosynthesis in these environments is influenced by factors such as temperature, nutrient availability, and water clarity. In contrast, terrestrial photosynthesis demands adaptations to cope with variable light conditions and atmospheric influences. Plants have evolved diverse leaf structures and orientations to maximize light absorption while minimizing water loss. The presence of stomata, small openings on leaf surfaces, regulates gas exchange, allowing for the uptake of carbon dioxide while controlling transpiration. In arid regions, succulents exhibit specialized mechanisms to conserve water, showcasing the remarkable diversity within terrestrial photosynthetic strategies. Both systems engage in the fundamental process of converting light energy into chemical energy, yet the pathways and efficiencies differ. Aquatic environments often demonstrate rapid turnover rates, while terrestrial systems can exhibit longer life cycles. These variations highlight the intricate balance of life forms within their respective ecosystems, revealing the delicate interplay of light, water, and nutrients that sustains life on Earth. And ibigwain duagansam Earths of life forms within their respective ecosystems. Carbon dioxide is fixed directly into a 3-carbon compound through a series of reactions known as the Calvin cycle. This process occurs in the chloroplasts of cells, predominantly in temperate regions, where conditions are often conducive to efficient photosynthetic activity. It relies heavily on ambient carbon dioxide levels, which can limit efficiency during periods of drought or high temperature. In contrast, C4 photosynthesis introduces an additional step before carbon dioxide fixation, where it is first incorporated into a 4-carbon compound. This adaptation enables certain plants to capture carbon dioxide more efficiently, particularly in hot, arid environments. This method minimizes photorespiration, a process that can waste energy and reduce overall productivity. C4 plants, such as maize and sugarcane, showcase an evolutionary response to climatic challenges, allowing them to thrive where C3 plants may falter. The structural differences between these pathways, including specialized leaf anatomy and cellular compartmentalization, reveal the remarkable adaptability of plant life. This adaptation not only influences the growth and productivity of individual species, but also shapes entire ecosystems, affecting biodiversity and agricultural practices across various regions of the globe. In arid environments, the necessity for survival has led to the evolution of CAM photosynthesis, a remarkable adaptation. This process allows certain plants to conserve water by opening their stomata during the night rather than during the heat of the day. The uptake of carbon dioxide occurs in the cooler hours, reducing water loss significantly. The stored carbon dioxide is then utilized during daylight for photosynthesis. Succulents and cacti exemplify this remarkable adaptation, thriving in climates where moisture is a limiting factor. Their thick, fleshy tissues store water, enabling them to endure prolonged periods of drought. This physiological strategy not only enhances their resilience, but also contributes to the stability of local ecosystems. In these arid regions, the presence of CAM plants influences the distribution of other species. The ability to draw carbon dioxide at night allows them to coexist with more traditional flora, which rely on the C3 pathway. This coexistence can create a diverse array of microhabitats, supporting various life forms adapted to the unique conditions. Soils in these areas often display specialized nutrient cycles, as the organic matter from decaying cam plant material enriches the ground, fostering a delicate balance. Such dynamics underscore the intricate relationships within ecosystems, revealing nature's capacity to adapt and thrive amidst adversity. The evolution of photosynthesis marked a pivotal shift in the development of life on Earth. Initially, early organisms relied on anaerobic processes, extracting energy from organic compounds without utilizing oxygen. This method sustained primitive life forms, allowing them to flourish in an oxygen-poor environment. As these organisms proliferated, they began to harness sunlight, leading to a gradual transition towards more sophisticated energy acquisition. For the emergence of cyanobacteria served as a crucial turning point. Through the process of photosynthesis, they converted light energy into chemical energy, releasing oxygen as a byproduct. This event, known as the Great Oxidation Event, transformed Earth's atmosphere. The accumulation of oxygen introduced new possibilities for life, enabling organisms to evolve aerobic metabolism, which is significantly more efficient than its anaerobic counterpart. In this region of scientific inquiry, the adaptation of organisms to utilize oxygen allowed for a diversification of life forms. Subsequently, aerobic respiration became the dominant energy-producing process, supporting complex multicellular organisms. The interplay between photosynthesis and respiration illustrates a delicate balance within ecosystems, where the byproducts of one process fuel the other. Through this evolutionary lens, the development of photosynthesis not only underscores the ingenuity of life, but also highlights the profound interdependence of biological systems on this planet. The emergence of cyanobacteria marks a pivotal moment in Earth's history, representing the earliest known photosynthetic organisms. These microscopic entities, thriving in ancient oceans, harnessed sunlight to convert carbon dioxide and water into organic compounds. This process, imbued with simplicity yet profound implications, led to the release of oxygen as a byproduct, reshaping the atmosphere over millions of years. Once confined to primordial waters, these organisms played a crucial role in the Great Oxygenation Event. As cyanobacteria proliferated, the oxygen they released began to accumulate in the atmosphere, leading to significant changes in environmental conditions. The gradual increase in oxygen levels allowed for the emergence of aerobic life forms, fundamentally altering the trajectory of biological evolution. In addition to oxygen production, cyanobacteria exhibited remarkable adaptability, colonizing diverse habitats. Their ability to form symbiotic relationships with other organisms paved the way for the evolution of more complex life. This adaptability ensured their survival through various geological epochs, including periods of mass extinction. Current research continues to explore the genetic and metabolic pathways of these ancient organisms, revealing their significance beyond mere oxygen producers. Understanding cyanobacteria enriches knowledge of Earth's past, while providing insights into potential applications in biotechnology and ecological restoration. Their legacy remains integral to the ongoing narrative of life on this planet. The intricate relationship between chloroplasts and their ancestral origins reveals a fascinating chapter in the evolution of photosynthesis. This theory posits that these organelles descended from free-living cyanobacteria, which established a symbiotic relationship with ancestral eukaryotic cells. Over time, this union facilitated the transformation of how energy is harnessed in living organisms. The engulfment of cyanobacteria marks a pivotal moment in cellular evolution. Once incorporated, these photosynthetic entities began to operate within their new host, converting sunlight into chemical energy through the process of photosynthesis. This development allowed for the production of oxygen as a byproduct, significantly altering the atmosphere and paving the way for diverse life forms. Significantly altering the atmosphere and paving the way for diverse life forms. Chloroplasts, now integral to plant cells, retain remnants of their prokaryotic lineage. They possess their own circular DNA reminiscent of bacterial genomes, along with ribosomes that closely resemble those found in cyanobacteria. Such features underscore the enduring legacy of their evolutionary past. This evolutionary transition not only catalyzed the advent of oxygen-rich environments, but also led to the establishment of complex ecosystems. In this context, the role of chloroplasts transcends mere functionality, embodying a profound connection between light and life that continues to shape the biosphere today. The interplay of these processes remains a cornerstone of biological existence, influencing both climate and ecological dynamics. The emergence of photosynthesis marked a pivotal moment in Earth's history, leading to the gradual accumulation of oxygen within the atmosphere. This process, primarily carried out by cyanobacteria and later by plants, transformed sunlight into chemical energy, producing oxygen as a byproduct. Over billions of years, the oxygen released began to permeate the oceans and the atmosphere, reshaping the planet's environmental landscape. As this gas accumulated, it facilitated the development of aerobic organisms, which thrived in oxygen-rich environments. The rise in atmospheric oxygen levels catalyzed a series of evolutionary adaptations, allowing for more complex life forms to emerge. This transition was not instantaneous. Rather, it occurred in phases, characterized by significant geological and biological milestones. The Great Oxidation Event, occurring approximately 2.5 billion years ago, serves as a key marker in this evolution. It represents a time when oxygen levels rose dramatically, leading to widespread extinction of anaerobic organisms, which could not survive in the new conditions. This shift laid the groundwork for the diversification of life. As these processes unfolded, they altered weather patterns, climate systems, and even the chemical composition of the oceans. The oxygenation of the atmosphere remains a testament to the intricate relationship between life and the planet's geochemical cycles. The role of photosynthesis in the carbon cycle is foundational, establishing a vital link between the biosphere and the atmosphere. Through this process, carbon dioxide is absorbed by green plants, algae, and certain bacteria, transforming it into organic compounds essential for life. This action serves not only to sequester carbon, but also to release oxygen, which is critical for aerobic organisms. In this region, chlorophyll-containing organisms capture sunlight, using its energy to convert carbon dioxide and water into glucose and oxygen. The glucose produced becomes a primary energy source, fueling growth and reproduction. As living organisms metabolize this energy, they release carbon dioxide back into the atmosphere, completing a cycle that sustains life on Earth. Decomposers play a crucial role in this process as well. When organisms die, their bodies are broken down by bacteria and fungi, returning carbon to the soil and atmosphere. This decomposition contributes to the reservoir of carbon stored in organic matter, influencing soil fertility and the overall health of ecosystems. The delicate balance maintained by photosynthesis and respiration underpins the carbon cycle, regulating atmospheric carbon levels and impacting climate. Changes in these processes can lead to significant shifts in global climate patterns, affecting weather systems and biodiversity across the planet. Alterations in photosynthesis not only influence climate, but also determine the resilience of ecosystems. The ability of plants to absorb carbon dioxide and release oxygen serves as a fundamental mechanism for maintaining atmospheric balance in regions where photosynthetic efficiency declines, such as areas suffering from prolonged drought or increased temperatures. The impacts ripple through the food web. Declining plant health reduces available oxygen and alters the composition of habitats, leading to diminished biodiversity. In response to these changes, certain species exhibit remarkable adaptations. Some plants develop deeper root systems to access water reserves, while others may alter leaf structures to minimize water loss. These adaptations allow for survival in increasingly hostile environments. Shifts in flowering times and fruiting cycles also reflect the need for plants to align with changing climatic conditions, ensuring reproductive success despite the challenges posed by climate change. Marine photosynthetic organisms, such as phytoplankton, also undergo transformations. Elevated temperatures and ocean acidification affect their growth and distribution, impacting the entire marine ecosystem. These microscopic producers are essential for oxygen production and carbon sequestration, making their adaptation vital for ocean health. Collectively, these processes underscore the intricate connections between photosynthesis, climate change, and the delicate balance of life on Earth. Understanding these dynamics is essential for developing strategies aimed at mitigating climate impacts and fostering ecosystem resilience. The relationship between photosynthesis and respiration forms a continuous cycle that sustains life on Earth. This process begins with photosynthesis, where plants, algae, and certain bacteria convert sunlight into energy, transforming carbon dioxide and water into glucose and oxygen. The oxygen released becomes essential for aerobic organisms, including humans, and the glucose serves as a vital energy source for growth and metabolisms. In contrast, respiration occurs in all living organisms, where glucose is broken down to release energy stored during photosynthesis. This process involves consuming oxygen and producing carbon dioxide and water, effectively reversing the photosynthetic equation. The carbon dioxide emitted during respiration re-enters the atmosphere, where it can again be utilized by photosynthetic organisms, thus maintaining the balance between these two processes. This intricate exchange illustrates the interdependence of life forms within ecosystems. In densely forested regions, for instance, the high rates of photosynthesis contribute significantly to atmospheric oxygen levels. Conversely, respiration in these regions facilitates the recycling of organic matter, supporting nutrient cycles. The efficiency of this relationship varies with environmental conditions, such as light intensity, temperature, and availability of water and nutrients. These factors contribute significantly to the establishment of food chains, where the process of photosynthesis serves as a foundational element. Organisms that perform photosynthesis, primarily plants, algae, and certain bacteria, convert solar energy into chemical energy. This transformation not only sustains their own growth, but also supports an intricate web of life. The energy captured during this process is stored in the form of glucose, which serves as a vital source of nourishment. Herbivores depend on these primary producers for sustenance, deriving energy directly from the plants that harness light. In turn, carnivores rely on herbivores for their energy needs, further illustrating the interconnectedness within ecosystems. This flow of energy from the sun through photosynthetic organisms to various levels of consumers underscores the importance of photosynthesis in maintaining ecological balance. Decomposers also play a crucial role. breaking down organic matter and returning essential nutrients back to the soil, where they can be reabsorbed by plants. Without this continuous cycle of energy transfer and nutrient recycling, ecosystems would struggle to thrive. The efficiency of photosynthesis not only fuels individual organisms, but also sustains entire habitats, impacting biodiversity and the stability of the environment. In this way, the process underpins not just life itself, but the very structure of ecological communities. Photosynthesis serves as a cornerstone for biodiversity, shaping the intricate web of life within ecosystems. This process fuels the growth of plant species, which in turn provides habitat and sustenance for a myriad of organisms, from towering trees to microscopic phytoplankton. The diversity of photosynthetic life forms creates a foundation for ecological interactions. The varied plant life enables different ecological niches, supporting herbivores that graze upon them and predators that rely on those herbivores. Each species plays a role in maintaining the balance necessary for ecosystem resilience. In these complex networks, competition, cooperation, and symbiosis unfold, driven by the energy harnessed through photosynthesis. The benefits extend beyond individual organisms, encompassing essential ecosystem services. These include carbon sequestration, which mitigates climate change, and soil stabilization, which prevents erosion and promotes fertility. Pollination, facilitated by numerous species attracted to flowering plants, ensures the reproduction of many crops and wild plants alike. Additionally, the variability within photosynthetic organisms contributes to genetic diversity, which is crucial for adaptation to environmental changes. The myriad forms of life that arise from this single process highlight the interconnectedness of ecosystems, illustrating how the health of one component is reflective of the overall well-being of the environment. This intricate relationship underscores the importance of protecting photosynthetic systems, as they are vital for sustaining not only biodiversity, but also human existence itself. The role of photosynthesis extends into the realm of human agriculture, providing the foundation for food production across the globe. This process enables plants to convert sunlight into chemical energy, forming the basis of the food chain. Grains, fruits, and vegetables, all products of photosynthesis, serve as essential nutrients for both humans and livestock. In cultivated fields, the efficiency of photosynthesis directly influences crop yields. Optimal sunlight, adequate water, and nutrient-rich soil create ideal conditions for this crucial biological function. Advances in agricultural science aim to enhance these conditions, employing techniques such as selective breeding and genetic modification to increase the rate of photosynthesis in key crops. These methods seek to address the growing demand for food as populations expand. Climate change poses significant challenges, altering weather patterns and impacting the availability of essential resources. Such disruptions can diminish the effectiveness of photosynthesis, threatening food security. Research continues into sustainable practices that can bolster resilience against these changes. Agroforestry, crop rotation, and the use of cover crops are strategies that enrich the soil and promote biodiversity, thereby enhancing the overall health of ecosystems. Through these methods, the delicate balance maintained by photosynthesis can be supported, ensuring that agriculture remains viable in a changing environment. Technological advances have transformed the ability to measure photosynthesis, providing researchers with unprecedented insights. Innovative instruments, such as gas exchange analyzers, allow for real-time monitoring of carbon dioxide uptake and oxygen release in plants. These devices, equipped with sensitive sensors, can detect minute changes in gas concentrations, unveiling the intricacies of photosynthetic efficiency under various conditions. Remote sensing technologies, utilizing satellite imagery and aerial drones, facilitate large-scale assessments of vegetation health. By analyzing spectral reflectance, scientists can infer chlorophyll content, indicating the photosynthetic capacity of vast agricultural landscapes. This approach enables monitoring over time, revealing patterns and trends that guide sustainable practices. Fluorometers have introduced another layer of understanding by measuring chlorophyll fluorescence. This technique assesses the energy conversion efficiency of photosystem 2, directly linking light absorption to photosynthetic output. Such data contributes to a deeper comprehension of plant responses to environmental stresses, such as drought or nutrient deficiency. Artificial intelligence and machine learning algorithms further enhance data analysis, enabling the interpretation of complex photosynthesis-related datasets. These technologies can predict how plants will respond to future climate scenarios, thus aiding in the development of resilient crops. With each advancement, the synergy between technology and plant science deepens, fostering a more sustainable relationship with the environment. This evolving landscape promises to unlock new avenues in optimizing photosynthesis for future generations. Remote sensing techniques have emerged as invaluable tools in monitoring photosynthesis across diverse ecosystems. These methodologies rely on satellite imagery and aerial data collection to assess vegetation health and productivity over vast areas. Through the analysis of light reflectance, specific wavelengths associated with chlorophyll can be detected, providing insights into the efficiency of photosynthetic processes. The Normalized Difference Vegetation Index, or NDVI, serves as a critical metric, quantifying the greenness of vegetation. This index highlights changes in photosynthetic activity, allowing for the assessment of plant health and stress levels. By capturing data across different seasons, it becomes possible to observe how environmental variables influence photosynthesis in real time. Multispectral and hyperspectral imaging systems further enhance the understanding of photosynthetic dynamics. These technologies can distinguish subtle variations in leaf chemistry and structure, revealing how plants adapt to their surroundings. The integration of remote sensing with ground-based measurements creates a comprehensive picture of photosynthetic activity, bridging the gap between localized studies and broader ecological trends. These techniques not only aid in agricultural practices, but also contribute to monitoring global carbon cycles. Understanding the efficiency of photosynthesis on a larger scale is crucial for informing climate models and predicting the impacts of environmental change on food security and ecosystem health. Artificial photosynthesis seeks to replicate nature's intricate process, aiming to transform sunlight into chemical energy. This innovative approach harnesses solar energy, converting it into fuel through synthetic means. Researchers are exploring various materials and methods to effectively imitate the efficiency observed in natural systems. Catalysts, often derived from abundant materials, play a pivotal role in these processes. They facilitate the conversion of carbon dioxide and water into carbohydrates and oxygen, much like plants do. By optimizing these catalysts, scientists strive to enhance the energy conversion efficiency, potentially surpassing the limitations of natural photosynthetic pathways. The implications of successful artificial photosynthesis extend beyond energy production. It holds promise for carbon capture technologies, mitigating atmospheric carbon dioxide levels while generating usable fuels. This dual functionality aligns with global efforts to combat climate change, offering a sustainable pathway to energy generation. Research is also advancing in the realm of integration. Combining artificial systems with existing energy infrastructures could lead to more robust solutions for renewable energy storage. The synergy of these technologies may provide a means to address fluctuating energy demands while reducing reliance on fossil fuels. Genetic engineering has emerged as a pivotal tool in enhancing photosynthetic efficiency. By manipulating the genetic makeup of plants, scientists can optimize the pathways that convert sunlight into energy. This process often involves altering specific genes that govern the structure and function of chloroplasts, the cellular organelles where photosynthesis takes place. Modifications can lead to increased chlorophyll production, allowing plants to capture more light energy. In this region of research, One focus lies on improving the carbon fixation process by introducing genes from more efficient organisms, such as certain algae or bacteria. It is possible to enhance the conversion of carbon dioxide into organic compounds. These modifications not only increase yields but also promote resilience against environmental stresses. The integration of synthetic biology further expands potential avenues for improvement. By designing entirely new metabolic pathways, researchers can enable plants to utilize light more efficiently at varying wavelengths. This results in better growth rates and higher productivity, which is crucial for meeting the food demands of a growing population. Field trials have demonstrated promising results, revealing how genetically modified crops can outperform their conventional counterparts under specific conditions. As research continues, the implications for agricultural practices and global food security become increasingly significant. This exploration in genetic engineering not only highlights the complexities of life, but also reveals the intricate relationship between biology and technology. The pursuit of sustainable energy sources is increasingly turning attention towards biofuels, derived from the same process that powers photosynthesis. This biological mechanism, which allows plants to convert sunlight into chemical energy, offers a potential pathway to reduce reliance on fossil fuels. Integrating insights from photosynthesis into biofuel production could enhance efficiency and yield, aligning agricultural practices with energy goals. Advancements in genetic engineering provide opportunities to optimize crop strains, enabling them to absorb sunlight and convert carbon dioxide into energy more effectively. By enhancing the natural processes of certain organisms, researchers aim to develop biofuels that are not only renewable, but also capable of meeting the demands of a growing population. This approach emphasizes the potential of using algae and other microorganisms which can thrive on non-arable land and utilize waste products, thus minimizing competition with food crops. The implications of these developments extend beyond energy production. By utilizing biofuels derived from photosynthetic organisms, a reduction in greenhouse gas emissions is achievable, contributing positively to climate change mitigation efforts. This symbiotic relationship between energy and ecology is fundamental, as it challenges traditional views of resource consumption and promotes a vision of a more sustainable future. The ongoing research into these biological systems continues to illuminate pathways toward a cleaner, greener planet. Photosynthesis in extremophiles reveals a remarkable survival strategy adapted to harsh environments. These organisms thrive in conditions where conventional life struggles, such as high salinity, extreme temperatures, and intense radiation. In such locales, the process deviates from the familiar patterns observed in more traditional photosynthetic organisms. In saline environments, certain extremophiles utilize specialized pigments and biochemical pathways to capture light energy. These adaptations enable them to convert sunlight into chemical energy, even when faced with the osmotic challenges of their surroundings. High temperatures further push the limits of photosynthetic efficiency, as thermophilic species develop proteins that maintain functionality amid denaturation risks. In arid regions, moisture becomes a limiting factor for survival. Here, some extremophiles have evolved to harness light with minimal water requirements, optimizing their metabolic processes for survival. Their unique cellular structures enhance the capture of available light energy, further enabling their persistence in desiccated habitats. Radiation-resistant extremophiles employ specific pigments that shield them from harmful ultraviolet rays. These adaptations not only allow survival but also facilitate the conversion of light into energy in environments where others cannot endure. Such resilience provides insights into the potential for life in extreme conditions beyond Earth, suggesting that photosynthesis may be a universal mechanism for harnessing energy across diverse worlds. Each discovery offers a deeper understanding of life's adaptability in the cosmos. Microalgae play a pivotal role in global photosynthesis, contributing significantly to the planet's oxygen production and carbon dioxide absorption. These microscopic organisms inhabit diverse environments, from freshwater lakes to the vast expanses of the ocean, thriving under varying conditions. In these aquatic ecosystems, they harness sunlight, converting it into chemical energy through the process of photosynthesis. This transformation not only sustains their growth, but also supports a myriad of marine life. Through their photosynthetic activity, microalgae generate an estimated 50% of the Earth's oxygen supply, a vital component for the survival of aerobic organisms. Their efficient energy conversion mechanisms allow them to flourish in nutrient-rich waters, often leading to blooms that can dramatically alter local ecosystems. These blooms, while sometimes detrimental in excess, are essential contributors to global carbon cycling. The ability of microalgae to absorb carbon dioxide makes them a focus of research in climate change mitigation strategies. By sequestering this greenhouse gas, they help to regulate atmospheric composition. This unique capability highlights their importance not only in maintaining ecological balance, but also in potential applications for biofuels and sustainable energy sources. These organisms exemplify the intricate web of life, demonstrating how even the smallest entities can have profound effects on planetary health. The interplay between photosynthesis and other biogeochemical cycles reveals a complex network of interactions essential for sustaining life. Carbon, nitrogen, and phosphorus all engage in a delicate balance, influencing and being influenced by the process of photosynthesis. Through the absorption of carbon dioxide, it directly contributes to the carbon cycle, sequestering carbon in biomass and releasing oxygen as a byproduct. In this region, a nitrogen fixation occurs concurrently, allowing certain organisms to convert atmospheric nitrogen into forms usable by plants. This process, supported by photosynthetic activity, enriches the soil and promotes growth. The availability of nitrogen often dictates the efficiency of photosynthesis, creating a feedback loop that enhances productivity in ecosystems. Phosphorus, another critical element, also plays a crucial role. It is vital for energy transfer within cells, particularly in the form of adenosine triphosphate. Photosynthesis depends on phosphorus for the synthesis of chlorophyll and other biomolecules. The cycling of phosphorus through soil and water further connects various ecosystems, ensuring that energy flows through food webs. Interactions between these cycles illustrate a finely tuned mechanism, where disruptions in one can lead to cascading impacts throughout the others. This connectivity highlights the importance of maintaining ecological balance, as each cycle supports the overall health of the planet, emphasizing the significance of these processes in shaping life on Earth. Growth. The introduction of pollutants disrupts the delicate equilibrium that sustains photosynthesis. Chemicals such as nitrogen oxides and sulfur dioxide infiltrate the atmosphere, forming acid rain that alters soil chemistry. This alteration affects nutrient availability, creating a less hospitable environment for plant life. As photosynthetic organisms struggle, their ability to convert sunlight into energy diminishes, resulting in reduced biomass production. In urban areas, particulate matter settles on leaf surfaces, blocking sunlight from penetrating the chlorophyll-rich tissues. This obstruction inhibits the process of light absorption, further weakening plant growth and productivity. The consequences extend beyond immediate ecosystems. Diminished plant health leads to decreased oxygen levels and increased carbon dioxide concentrations, exacerbating climate change. Aquatic environments also experience the ramifications of pollution, particularly through nutrient runoff that causes algal blooms. These blooms can deplete oxygen in water, resulting in dead zones where aquatic life cannot thrive. The intricate relationships between organisms become strained as vital food chains are disrupted. The long-term effects of pollution on photosynthesis ripple throughout ecosystems, influencing biodiversity and resilience. Plants serve as foundational components of ecological systems, and their decline can lead to cascading effects, impacting everything from soil health to animal populations. Understanding these dynamics is crucial for developing strategies to mitigate pollution and protect the processes that sustain life on Earth. Photosynthesis operates at the foundation of life, serving as a primary mechanism by which carbon dioxide and light energy are transformed into organic matter. In oceanic environments, this process is predominantly conducted by phytoplankton and marine plants. These organisms absorb sunlight and utilize it to convert carbon dioxide into sugars, releasing oxygen as a byproduct. This vital function not only supports aquatic ecosystems, but also plays a significant role in global carbon cycling. Ocean acidification has emerged as a pressing concern, arising from increased carbon dioxide concentrations in the atmosphere. When carbon dioxide dissolves in seawater, it results in the formation of carbonic acid, subsequently lowering the pH of ocean waters. This shift in acidity poses a threat to marine photosynthesis, particularly among calcifying organisms such as corals and shellfish. The changing conditions affect their ability to produce calcium carbonate, essential for maintaining their structures. The repercussions of these alterations ripple through marine food webs, impacting biodiversity and the overall health of ocean ecosystems. Reduced photosynthetic efficiency among phytoplankton can lead to decreased oxygen levels and diminished food supplies for higher trophic levels. Understanding the interplay between photosynthesis and ocean acidification is essential for predicting future shifts in marine environments and ensuring the resilience of these critical ecosystems. In this complex ecological framework, the role of mycorrhizae emerges as a crucial factor influencing photosynthetic efficiency. These symbiotic relationships between fungi and plant roots enhance nutrient uptake, particularly phosphorus, which is vital for energy transfer within photosynthetic processes. This partnership fosters a more robust exchange of resources, allowing plants to optimize their light absorption capabilities. Mycorrhizal networks extend far beyond individual plants, connecting diverse species within the ecosystem. This interconnectedness facilitates not just nutrient sharing, but also enhances resilience against environmental stresses. Such resilience becomes vital in the context of fluctuating climate conditions where optimal photosynthesis can be compromised. The presence of these fungi can lead to improved water retention in soil, further supporting plant health during periods of drought. Research indicates that mycorrhizae can also influence the efficiency with which carbon dioxide is assimilated during photosynthesis. Enhanced root systems allow for greater access to water and nutrients, which in turn can increase overall biomass production. This symbiotic relationship demonstrates the intricate balance that exists within ecosystems, where the health of one component directly impacts the efficiency and sustainability of others. Understanding these subtleties sheds light on the broader implications for food security and ecosystem management, emphasizing the need for holistic approaches in conservation efforts. The relationship between photosynthesis and soil health is foundational to the vitality of ecosystems. Through the process of photosynthesis, plants convert sunlight into chemical energy, producing oxygen and organic compounds essential for life. This organic matter, when deposited into the soil as litter or root exudates, enriches it, fostering a diverse microbial community that thrives on these nutrients. Such microbes play a crucial role in nutrient cycling, breaking down organic materials and releasing essential minerals, thereby enhancing soil fertility. Healthy soil is characterized by its structure, which is influenced significantly by plant roots. These roots create channels that improve aeration and water retention, promoting a balanced habitat for various organisms. In turn, these organisms contribute to the breakdown of organic matter, reinforcing the cycle of nutrient availability. This interconnectedness illustrates the importance of maintaining both plant health and soil integrity. Conversely, disruptions in photosynthetic activity, such as deforestation or pollution, can lead to soil degradation. Erosion becomes prevalent, diminishing the soil's ability to retain moisture and nutrients. As soil health declines, so does the overall productivity of the ecosystem, potentially leading to diminished crop yields and increased food insecurity. Recognizing the interplay between photosynthesis and soil health reveals the necessity for sustainable agricultural practices, ensuring the resilience of ecosystems for future generations. Urban environments present unique challenges and opportunities for photosynthesis, particularly through the integration of green infrastructure. The presence of vegetation within cityscapes not only contributes to aesthetic appeal, but also plays a critical role in enhancing air quality and mitigating urban heat islands. Green roofs and vertical gardens, often found on high-rise buildings, harness sunlight effectively, transforming otherwise barren surfaces into vibrant ecosystems. These installations create microclimates that support diverse plant species, which in turn engage in photosynthesis, converting carbon dioxide into oxygen. This process significantly lowers ambient temperatures and helps combat urban pollution. The strategic placement of trees along streets and in parks further amplifies these benefits, offering shade and reducing energy consumption for cooling. Biodiversity thrives in these green spaces, fostering habitats that support birds, insects, and other wildlife. As the interconnectedness of urban life and natural processes becomes increasingly evident, the role of photosynthesis extends beyond individual plants. It contributes to the overall health of urban ecosystems, promoting resilience against climate change and enhancing community well-being. The success of these initiatives emphasizes the importance of integrating nature into urban planning. By recognizing the value of photosynthesis in urban settings, city dwellers can cultivate environments that nurture life while addressing pressing environmental challenges. These practices forge a path towards sustainable coexistence between human activity and the natural world. Understanding photosynthesis is essential for grasping the intricate connections within ecosystems. This process underlies the foundation of life on Earth, converting solar energy into chemical energy stored in glucose. Knowledge of how plants, algae, and certain bacteria harness sunlight illuminates the vital role these organisms play in supporting food webs and oxygen production. The educational significance extends beyond biology into various fields, including chemistry and environmental science. It provides insights into energy transfer, molecular interactions, and the biochemical mechanisms that sustain life. These lessons promote an appreciation for biodiversity and the delicate balance of ecological systems. In educational settings, engaging with photosynthesis fosters critical thinking about environmental challenges. Students learn about the impacts of climate change, deforestation, and pollution on plant life and, consequently, on all life forms reliant on these primary producers. Awareness of such interdependencies encourages future generations to advocate for conservation and sustainable practices. Additionally, the exploration of photosynthesis can inspire innovations in renewable energy. Understanding how nature captures and utilizes solar energy can inform advancements in solar technology, leading to more efficient energy solutions. This intersection of biology and technology exemplifies the potential for nature-inspired approaches to address pressing global issues. The knowledge gained from studying photosynthesis serves as a cornerstone for fostering environmental stewardship and sustainable development. The cultural significance of plants extends far beyond their biological functions. They embody ecosystems, represent life, and serve as vessels of tradition and identity. In various societies, plants have held sacred meanings, often intertwined with religious and spiritual beliefs. This reverence reveals a deep connection to nature, where foliage becomes a symbol of resilience and growth. Rituals and customs throughout history have incorporated flora, highlighting the essential role plants play in human experiences. Harvest festivals celebrate the bounty provided by crops, while healing practices often rely on botanical knowledge passed down through generations. These connections illustrate how integral plants are to cultural heritage, shaping practices, law, and even art. The process of photosynthesis itself has influenced philosophical musings and scientific inquiry. The transformation of light into energy resonates with concepts of renewal and the cyclical nature of existence in literature and folklore. Plants frequently serve as metaphors for hope, change, and the interconnectedness of life. Urban landscapes increasingly reflect this significance, with green spaces becoming sanctuaries for reflection and community. The incorporation of plants in architecture and urban planning demonstrates an awareness of their psychological and environmental benefits. As societies evolve, the enduring relationship with plants remains vital, underscoring their role in both ecological and cultural narratives. The process of photosynthesis is integral to ecological restoration efforts, serving as a cornerstone for rebuilding ecosystems through it. Plants convert sunlight into energy, facilitating the growth of vegetation essential for restoring habitats. This energy transfer initiates a chain reaction, supporting a diverse array of life forms that depend on these plants for sustenance and shelter. In degraded landscapes, the introduction of native flora is paramount. These species, adapted to their environments, utilize photosynthesis to reclaim and stabilize soil, preventing erosion and promoting nutrient cycling. The restoration of plant communities not only enhances biodiversity, but also enhances carbon sequestration, a crucial function in the fight against climate change. Healthy plant life creates microclimates, fostering conditions that benefit other organisms. Pollinators, herbivores, and predators all thrive in regions rich in photosynthetic activity. Their interactions contribute to the resilience of ecosystems, enabling them to withstand disturbances such as drought or invasive species. This process is not instantaneous. It requires careful planning and monitoring. Restoration practitioners assess local conditions, selecting appropriate species and employing techniques that enhance photosynthetic efficiency. Over time, as these ecosystems mature, the benefits of photosynthesis extend beyond immediate restoration goals, promoting long-term ecological stability and reinforcing the intricate web of life that sustains our planet. Indigenous knowledge contributes significantly to the understanding of photosynthesis, integrating centuries of observation and interaction with local flora and fauna. Traditional practices reveal insights into the adaptive strategies of various plant species, highlighting their resilience and responses to environmental changes. This knowledge encompasses not just the biological processes, but also the intricate relationships among species within ecosystems. Local communities have long recognized the importance of seasonal cycles, climate variations, and soil health. They possess a profound awareness of how these factors influence plant growth and productivity. For instance, The timing of planting is often aligned with lunar phases or specific weather patterns, which can enhance photosynthetic efficiency. These practices reflect a deep connection to the land, revealing insights into the cultivation of crops that optimize sunlight absorption and nutrient retention. The integration of indigenous perspectives with scientific methodologies enhances the understanding of photosynthesis on multiple levels. It underscores the value of biodiversity and the preservation of native plant species that have co-evolved with local ecosystems. Collaborative research initiatives increasingly seek to merge traditional ecological knowledge with modern science, fostering a holistic approach to environmental stewardship. Such partnerships generate comprehensive strategies for conservation and restoration, emphasizing the necessity of respecting and incorporating diverse worldviews in the pursuit of sustainable practices. The intricate interplay between traditional wisdom and scientific inquiry continues to illuminate the fundamental processes that sustain life on Earth. Photosynthesis represents a cornerstone of global food security, directly influencing agricultural productivity and ecosystem stability. This process, which allows plants to convert sunlight into chemical energy, forms the basis of the food web. It supports not only the flora that dominate landscapes, but also the fauna that rely on these plants for nourishment. The efficiency of photosynthetic pathways varies among species, with certain crops exhibiting heightened capabilities to capture light and convert it into growth. Innovations in agricultural practices have sought to optimize these processes, employing techniques that enhance light absorption and carbon dioxide fixation. This manipulation of variables can lead to greater yields, supporting the growing human population. In regions where food scarcity is prevalent, the role of photosynthesis becomes even more pronounced. The cultivation of crops with enhanced photosynthetic efficiency can mitigate hunger and bolster economic resilience. Research into genetically modified organisms often focuses on improving this fundamental process, aiming to increase resilience against climate fluctuations. Climate change poses a significant threat to photosynthesis, altering temperature, light availability, and atmospheric carbon dioxide levels. These changes can disrupt the delicate balance that underpins food production systems. Understanding and improving photosynthesis is therefore crucial for developing sustainable agricultural practices that ensure food security for future generations, making it a key area of ongoing research in science and agriculture. Global warming introduces a myriad of challenges to the delicate balance of photosynthesis. Rising atmospheric temperatures can alter the enzymatic processes that drive this essential reaction, impacting the efficiency with which plants convert sunlight into energy. Elevated carbon dioxide levels may initially enhance photosynthetic rates, yet this response is tempered by other stressors linked to climate change. As precipitation patterns shift, droughts become more frequent, depriving plants of necessary water resources. This region experiences a decline in soil moisture, which in turn affects plant vitality and their ability to photosynthesize effectively. In conjunction with this, increased temperatures can lead to heat stress, causing stomatal closure. This physiological response reduces carbon dioxide uptake, further constraining the photosynthetic process. The complex interplay of these factors can result in reduced crop yields, jeopardizing the delicate food supply chain. Additionally, shifts in plant phenology, such as flowering times, may disrupt established ecosystems, affecting pollinators and other interdependent species. As climates continue to change, the long-term implications on photosynthesis warrant careful examination. The resilience of various species under these new conditions may reveal potential adaptations, but the overall impact on global agriculture remains a critical concern.
Effective management and strategic interventions are essential to navigate these challenges, ensuring that plants continue to thrive in a warming world. Medicinal plants hold a significant role within the framework of photosynthesis, contributing not only to their survival but also to the well-being of countless species, including humans. These organisms utilize sunlight to convert carbon dioxide and water into glucose and oxygen, a foundational process that sustains life on Earth.
In this context, while metabolic pathways involved in photosynthesis facilitate the production of phytochemicals, compounds that possess therapeutic properties, this region of botanical diversity showcases how certain plants have evolved to harness light energy, subsequently synthesizing secondary metabolites. These compounds can exhibit anti-inflammatory, antimicrobial, antioxidant activities, providing natural remedies for various ailments. The intricate relationship between photosynthesis and the production of these beneficial substances underlines the importance of maintaining healthy ecosystems, and in many instances, traditional medicine relies on these plants, with various cultures utilizing their healing properties for centuries. The ongoing exploration of these natural resources continues to reveal potential pharmacological applications, offering a glimpse into the biochemistry that supports human health. Sustainable practices in harvesting and cultivating medicinal plants not only protect biodiversity, but also ensure the viability of these crucial systems. As challenges arise from climate change, safeguarding these plants and their intricate processes becomes paramount for both ecological balance and human well-being. Research into photosynthesis faces an array of future challenges that could impact its understanding and application. The increasing prevalence of elevated carbon dioxide levels affects plant growth patterns and photosynthetic efficiency. Some species may adapt to these changes while others falter, leading to shifts in ecosystem dynamics. Investigating how various plants respond to these altered conditions is essential for predicting future agricultural yields and biodiversity. Technological advancements in studying photosynthesis present new possibilities. Innovations such as genome editing could enhance specific traits, enabling plants to optimize photosynthetic processes. This area of research holds promise for developing crops that thrive in less than ideal conditions, ensuring food security amid a changing climate. Simultaneously, studying the interactions between photosynthesis and other environmental factors, such as temperature and humidity, is crucial. These elements can dramatically influence plant metabolism and growth rates, necessitating a comprehensive understanding of their interdependencies. The role of microorganisms in soil health and nutrient availability also intersects with photosynthesis research. These organisms contribute to the overall efficiency of nutrient uptake by plants, impacting their ability to photosynthesize effectively. Addressing these multifaceted challenges will require collaboration across disciplines, integrating ecology, genetics, and climate science to deepen the understanding of this essential biological process. Each step forward strengthens the foundation for sustainable practices and resilience in our ecosystems. Interdisciplinary approaches to studying photosynthesis reveal intricate relationships between biology, chemistry, and physics. This interconnectedness allows researchers to explore the molecular mechanisms that underpin this vital process. By utilizing tools from various scientific fields, insights emerge into how light energy is captured and converted into chemical energy within chloroplasts. Physics contributes to understanding the light harvesting complexes that efficiently capture photons. These complexes operate through resonance energy transfer, a phenomenon that allows for the rapid movement of energy, optimizing the efficiency of photosynthesis. This foundational knowledge informs agricultural practices by enhancing crop yields through engineered photosynthetic pathways. Anous som stast retaking salty, of the fenita when taus sophadelis, niurais secrodes the antavago sri, is meticulously studied through chemical kinetics, allowing scientists to understand the rates and efficiency of these reactions. This knowledge fuels innovations in carbon capture technologies, essential for addressing climate change. Biological research, on the other hand, examines how environmental factors influence photosynthetic efficiency, investigating the genetic and physiological traits of various plant species enables the identification of resilient varieties capable of thriving in changing climates. This collaborative framework fosters advancements in sustainable agriculture, ecosystem restoration, and climate adaptation, ensuring that the foundational processes of life continue to flourish. The contributions of citizen science to photosynthesis research have become increasingly significant. Enthusiastic participants engage in data collection, observing plant responses to environmental changes in their local habitats. This involvement generates an expansive dataset, capturing diverse conditions from urban gardens to remote wilderness areas. These observations offer invaluable insights into the adaptability of various species, revealing how they optimize their photosynthetic processes. Community-driven projects empower individuals to monitor plant health and growth, measuring factors such as leaf chlorophyll content and sunlight exposure. The amassed information serves as a bridge between professional scientists and the general public, fostering a deeper understanding of the intricate mechanisms of photosynthesis. It also allows for real-time tracking of changes in plant communities, identifying shifts that may signal broader ecological trends. Collaborative networks enable the sharing of findings, urban spun spice, enhancing the collective knowledge surrounding this essential biological process. Patterns discerned from citizen-contributed data can inform conservation strategies, guiding efforts to safeguard vulnerable ecosystems. The integration of amateur observations with formal research provides a comprehensive view of how plants respond to climate variability, enriching the overall narrative of photosynthesis. This synergy between professionals and volunteers not only advances scientific inquiry, but also cultivates an appreciation for the delicate balance of life sustained by photosynthetic organisms. The future of this research hinges on continued collaboration and engagement at every level. The potential for photosynthesis to support life beyond Earth presents a fascinating frontier. In the vacuum of space, where conditions are harsh and resources scarce, harnessing the power of this process could transform human exploration. Innovative experiments have already demonstrated that certain plants can thrive in controlled environments aboard spacecraft, utilizing artificial light to simulate sunlight. These environments, meticulously designed to replicate Earth-like conditions, allow researchers to monitor growth patterns and nutrient uptake. The study of how plants adapt to microgravity reveals their remarkable resilience. Changes in root orientation and leaf structure provide insights into the fundamental mechanisms that govern life. In this context, understanding the intricacies of photosynthesis becomes paramount. The efficiency of converting light energy into chemical energy influences not only plant health, but also the sustainability of life support systems. Plants recycle carbon dioxide and produce oxygen, essential for human survival during prolonged missions. Research facilities on the International Space Station have become laboratories for growing crops like lettuce and radishes, showcasing the viability of space agriculture. The cultivation of these plants not only serves nutritional purposes, but also contributes to the psychological well-being of astronauts, offering a glimpse of green life amidst the metallic confines of spacecraft. With continued advancements, this exploration could pave the way for future colonies on Mars and beyond. The intricate relationship between life and light forms a profound foundation for understanding existence itself. Photosynthesis serves as a bridge, uniting the vibrant energy of the sun with the essential processes of life. This transformation of light into chemical energy not only sustains individual organisms, but also underpins entire ecosystems. In essence, it creates the very fabric of life on Earth. The implications of this process extend far beyond biology. They provoke contemplation regarding the interconnectedness of all living beings. Each organism, reliant on sunlight, participates in a vast, interdependent web. The energy harnessed through photosynthesis fuels not only plant life but also herbivores, and are subsequently their predators. This cyclical flow of energy highlights a delicate balance where the survival of one is intricately linked to the existence of another. Examining photosynthesis further invites questions about the nature of intelligence and adaptation. Organisms have evolved diverse strategies to optimize light absorption, reflecting an innate understanding of their environment. This phenomenon raises inquiries into the consciousness within the natural world, urging a re-evaluation of what it means to be alive. On contemplating the role of light as a catalyst for life, a deeper appreciation emerges for the symbiosis inherent in the universe. Such reflections illuminate the significance of sustainability and the shared responsibility of preserving this delicate balance. The continuing journey of understanding photosynthesis unfolds in layers of complexity and beauty. Researchers delve into the intricate mechanisms that allow organisms to harness sunlight, converting it into energy with remarkable efficiency. This process, rooted in chlorophyll's capacity to absorb specific wavelengths of light, enables plants to thrive in diverse environments, from lush rainforests to arid deserts. As investigations progress, insights into the evolutionary adaptations of various species reveal how life on Earth has optimized this fundamental process. The interplay between light, water, and carbon dioxide creates a dynamic system that supports not only plant life, but also the myriad organisms that depend on them for sustenance. These relationships illustrate the interconnectedness inherent in ecosystems. Recent advancements in technology allow for deeper exploration of photosynthesis at the molecular level. Scientists employ sophisticated imaging techniques and genetic engineering to unlock the mysteries behind this vital process. Understanding these mechanisms paves the way for innovative agricultural practices, enhancing crop yields and promoting food security. The implications extend beyond earthly boundaries. Knowledge gleaned from photosynthesis may inform strategies for sustaining life in extraterrestrial environments, where similar principles could be applied. This pursuit of understanding signifies a commitment to safeguarding the biosphere, ensuring that the alchemy of turning light into life continues to flourish for generations to come.